Hello, good afternoon, and welcome back for this last session of the Human Cell Atlas Latin America. In the last session, you go to show to you the setup that you just have done, and you just set up a 10x machine here in Brazil. It's the first operational machine in Brazil. If I'm not wrong, I mean, it's the third 10x machine running in Latin America. And in order to do this, we are very lucky. I mean, just got back from Canada. Our colleague Camila Fernandes Nascimento, they come back from Canada when she had the opportunity to set up the technology in her pharma lab. Plus, Camila, I want to welcome you, and if you can tell Daniel what to go to show for us today.
Sure. First of all, I would like to thank the organizers and also Lucio for this amazing opportunity. So as he was telling us, I had this incredible opportunity of working with the single cell technology in Canada. So I work at Gustavo Turek's lab, and they have a brain bank there. So they work with postmortem brain samples. So I had this great opportunity to optimize the protocol for the single-nuclei ATAC-Seq in his lab. And I just came back and we have the machine here. So I'm very excited to optimize protocols here so we can all start using this machine. And now we are sharing with you a video with some technological aspects of the technology that we are using. And then also I speak a little bit about my experience in Canada, and afterwards we're gonna see a video where we show you step by step the protocol for this Chromium Controller machine that we just received. So I hope you enjoy, and then afterwards we will be waiting for questions. We'll be happy to answer the questions. Thank you. Hello everyone. I'm Camila Nascimento. I am a postdoc at the University of São Paulo Medical School in Brazil. Last year, I had this incredible opportunity to implement the single cell technology in a laboratory in Canada. And now we are seeking to implement this technology in Brazil, in São Paulo. So, and this, we are working together, me and Lucio, to make this happen, to implement the single cell technology in Brazil. So, as we all know, we all scientists are spending years of our lives to try to classify the cells by their structure, functions, locations, and more recently, molecular profiles. But the characterization of cell types and states, it remains surprisingly limited. So, like as we all have been discussing here during this amazing workshop from the Human Cell Atlas, we are all interested in have a more in-depth analysis of the cells and their state, and with that, the molecular states within tissues and systems in the full diversity, which we are now able to do thanks for this emerging transformative advances in experimental and computational methods, and this leads us to the single cell technology. So, like a very nice analogy that we can make is the smooth analysis. So before, like we used to analyze our samples as bulk and as like a smoothie. So here's like our tissue with its specific parts, so its specific cell types and states, and different cell states and physiology and molecular biology. And when we were studying the tissue, for example, we were analyzing like all the cells together, like a smooth, And now with the single cell technology, we can separate the components. And for this, we can, for example, know, oh, for example, is this yogurt? What probiotics are in this yogurt, or the fruit, or for example, are the raspberries organic? So we can go more in depth on the cell states, and not only like it's. And we can go more in depth in the cell state. So, and then like with this amazing technology of the single cell and the sequencing platforms, as well as the bioinformatic tools, we have been able to further understand the cellular states and heterogeneity. And this is relatively a new field, especially for ATAC-Seq. So these are like all the techniques that have been developed for us to look at single cell data. And the gray dots, they are corresponding to the RNA-Seq techniques. And the black dots are the ATAC-Seq studies that have been published. So we can see that for RNA-Seq, it started around 2009, 2010. And it's amazing like how the types of platforms that we have available are increasing. And around 2015, 2016, we have also like published studies on ATAC-Seq using ATAC-Seq technology. And we'll discuss a little bit more about this, but then what can we see? Like, what does this techniques provide us? So, for example, for the single-cell RNA-Seq, we can identify and characterize rare cell types and biomarkers. So we can analyze and understand how cellular heterogeneity contributes to biological system, examine therapeutic mechanisms of action on a cell-by-cell basis, increase resolution of transcriptionally similar cell types with protein information, and we can discover cellular heterogeneity stemming from epigenetic variability. This is like mostly with what ATAC-Seq does, and this is like mostly what RNA-Seq does. And better understand the gene regulatory networks that are upstream of gene expression. We can define cell types and states for lineage and developmental program tracing. And with that, we can also accelerate biomarker discovery. So there are several techniques that I just show you, like we have different types of techniques, but for us in our lab, we are implementing one of them, and it's the 10x genomics, from the 10x genomics, and they have this very nice system that they use this machine. It's called Chromium Controller, and we are implementing this machine in our facility, the Phenotypic Screening Platform Institute of Biomedical Science at the University of São Paulo. And with this machine, with this controller, with these techniques from 10x, we can do different types of cellular solutions. For example, single cell gene expression, single cell ATAC, single cell immune profiling, single cell multi-omic ATAC plus genomic expression, among others. And the workflow for this technology is basically first you do your sample preparation, fresh, and you can use for that either fresh or cryopreserved tissue. And of course, we need to remember like all the we need to follow all the ethical guidelines that you have in your institution. And the second step is doing the single cell separation and library construction. And this technology is the same base for all the analysis, all the ATAC solutions, all the single cell solutions, sorry, that you have. So, and after you separate your cells and you do your library construction, you sequence your data, you do your data analysis, then you need bioinformatic tools for that. And you can also visualize your data, and the same company provides you with a software that's more friendly for people that aren't, for researchers that are not bioinformaticians. And it's nice because you can like see the clusters of your cells and analyze and try to assign cell types according to the markers that you found in that cluster. So it's a very nice tool that you can use it as well. But for this presentation, we are focusing on the sample preparation and the single cell separation and library construction. So the workflow for us here is first you have your 10x barcoded gel bits. So in each of these gel bits, you have like a read one sequence and your 10x barcode. So your barcode, it's like a 16 long nucleotide. It's a sequence that will tell you exactly that this is like the same cell. So so this gel bits contains 3.5 million bit types with different barcodes. So then you can generate in a droplet and all the cells that are in the same droplet that I'm going to show you later, they are going to have the same barcode. And you also have your poly-D sequence here. And this is like for gene expression. And I'm also going to show you the ATAC that is the one that I had the opportunity to work with. So you have your cells and the enzymes to do the reverse transcription. And you have your gel bits with your 10x barcode, and you input in the chip that I'm going to show you. And then you have the oil, and the oil partition with the cell bit and the cell or nuclei. It depends on what you're doing. We call this a gen. So this is called the gen. So what is the gen? The gen, again, is the single cell, and it's the gel bit, and is the partitioning oil. And here in this space, we also have the reagents for the reverse transcription reaction. And then, so inside the machine, all this magic is happening. And then you also have the reverse transcription happening inside the genes, the gems. So you have within a gen, so inside one gen, you have your cDNA, your 10x barcoded, your barcoded DNA, cDNA. And then you amplify it, and then you sequence it. So this is basically what you do. So what you're gonna have, it's all cDNA generated in a droplet, carry an identical barcode, and the barcodes enable grouping the molecules as derived from the same droplet cell. And this is the machine. It's like a very small machine like this, and it's very easy to use. And you have the chip that you buy it, and the chip type, it depends on what technique you're gonna use. So, and then you have, but for all of them, you're gonna have your sample wells, you're gonna have your gel bit wells, and you're gonna have your recovery wells. So, and here you're gonna add your oil, and then here you're gonna have your gems and with the cDNA, barcoded cDNA inside. And important points that I would like to share with you is that the machine is very easy to use, and it takes it takes a little time to be ready. So all this process, it takes like less than 10 minutes it lasts it lasts 10 minutes. And for the single cell RNA, of course, it's important for us to have in mind that if you have like a good quality samples, obviously it's gonna reflect the quality of your barcoded cDNA. And for the cryopreserved tissue, one important point that I'm gonna share with you in the next slides is the quality of the nuclei extract. Like you need to have a nice nuclei, so you need to have a good protocol for nuclei extraction, and you also need like a good nuclei suspension. Because if you don't have, if your cells are aggregated, then you're gonna have duplicates. So instead of having like one cell here, you're gonna have 2 or 3, and then you're not having your single cell data accurately. And then one important point is that if you have your sample and you need like to ship somewhere else to do like the single cell separation, like for the company, it's not recommended that you like freeze it. So once you extract your nuclei, it's very nice that you can immediately separate and start with the formation of your gens and so on. So it is nice that you extract your nuclei, resuspend in your nuclei suspension solution, and then start right away the whole process of cell separation in the Chromium Controller. So this is kind of a downside if you are like far away from the place from the lab that has the machine. But at the same time, if you work with a cryopreserved sample, you can kind of like carry with you for the lab that you're gonna run your analysis, and then afterwards you can extract your nuclei and start the nuclei separation right away. So I'm saying like I'm sounding confused when I say nuclei or cell, because my experience was with post-mortem brain samples, cryopreserved post-mortem brain samples, and with these samples we needed to work with the nuclei, because it's not possible for us to get an intact cell. And for ATAC, this is actually very nice, because we are only we are interested actually in the chromatin. But for the RNA, so there are like some people, like some reviewers that would ask you that, oh, but you are not looking at the RNA, mRNA that are in the cytoplasm, you're looking at the RNA that are only inside the nuclei. So it's like a downside for the RNA. If you're working with cryopreserved samples, then you need to extract the nuclei only. So, but for the ATAC, this is not a problem, because you just need your nuclei. So what is the ATAC? So once, like we talked about gene expression, and it's more like intuitive for us, but then ATAC, not everyone is familiar with. So ATAC is the assay for transposase accessible chromatin with high throughput sequencing. So basically what this technique does is like you use this enzyme, it's called the TN5 transposase, and you use this enzyme. This enzyme, it's already contains the adapters. So this enzyme cuts the DNA and inserts the adapters at the end of this fragment, and this enzyme only cuts the DNA in the open regions of the DNA. So that's why you can study the accessible regions of the DNA. And then you amplify it and you sequence it. And again, one of the things that you can answer using this technique is cellular heterogeneity, gene regulatory networks, and cell lineage and developmental program tracing. And as I told you, I worked with postmortem brains. So the nuclei isolation that we used was the nuclei isolation protocol that I used was the one that the lab had already established. But the company also provides us with a suggestion of protocol that might work very well. So, but for me, like I had this, I used the protocol they had already established there. But then I'm sure that the one that is provided by the company might work very well. And then you isolate your nuclei and then you count your nuclei. So this step is very important because then so you're gonna set a concentration of cells that you input and the concentration of cells, and then you're gonna target a number of cells based on the concentration of cells that you loaded. And then this is important for your sequence step because you're gonna say, oh, I'm gonna target sequencing 5,000 cells, and for this I need this concentration to input, and then I'm targeting this much of cells, and then I'm gonna sequence this much of cells and number of reads that you want for each of the cells. So, and then you do the transposition that is made in bulk. So you treat your samples with this enzyme that cuts the DNA in the accessible regions. And these regions, and this occurs in bulk. And then afterwards, you include in the machine, and then you do the gem generation, same as I explained for the RNA data. And then you do your cleanup and your QC, your quality control, library construction, and cleanup again, and then sequencing it. So this is like, this is the gel bits for the ATAC. They are a bit different from the RNA, from the gene expression ones. But then again, you have your barcode that will identify the cell, the specific cell that are inside the gene. So again, you have your gel bits, you have your nuclei, your enzyme, your oil, and you input here in that shape that I showed you before. And then in each of the gem, we have so the cell, the gel bits, and the oil, the partitioning oil. And inside the gene, there is like this elongation of the fragments, of the DNA fragments that were cut, and also the barcode, the formation of the barcode. And then you amplify this, and so again, like the workflow, so you amplify and then you sequence and you do your analysis. So just for us to have an idea of the analysis, because some people like usually ask about it. So so we have your cell sample or your nuclei sample. It depends on what you're asking or your research question. And then you have your Chromium chip, and you have your gem. There are occurring inside your chip. And then you have your library. You sequence it, and then you can go through the cell ranger pipeline. That this is a pipeline that the company also provides you to do your data analysis. But of course, I think it's something that is flexible that you can adapt for your research interest. And then like during like this analysis, what it's done, it's like the alignment to the human genome, the deduplication, because you have to, so this is an important step for any sequencing data. And you also remove the mitochondria reads, and you identify the transposase cut sites. So just to give you an idea of how important the sample preparation step is. So I had to optimize this for this post-mortem brain sample. So before, this is how the cells looked, the nuclei actually look at before the optimization, and this is how it looked like after optimization. So we needed to optimize the concentration of cells that we were suspending in the nuclei buffer. And this is what was a critical step for us. It sounds very simple, but if you don't go over it step by step, then you cannot know. And this here, you can see like a lot of cellular debris, and you can also see like some some cells that are aggregated, and this is something that we don't want to see. So it's very nice that you can look at the nuclei and the cells before you input in the machine. And this is like I would say that it could be like a quality control step. And also, like for the ATAC-Seq data, because of the nucleosome pattern that we have for the transposase cut sites, so the library, the perfect library usually looked like this because of the nucleosome patterning. And this is like how our library looked like on our first trial, and this is our library after optimization. So you can see that the library is more concentrated, and the bumps are exactly how we expect. And also like and also it makes a lot of difference on your results. So here you can see like the cell, so each dot, it corresponds to a cell. So this is like our first trial. You can see that our library was not very well, it was not good, didn't have a good concentration, and also we couldn't see like a lot of clusterization after the analysis. And this is after optimization. So each dot that you see is a cell type, and this is a tCNA graph showing that the clusters that basically show like phenotypically, they are phenotypically similar. So then they form this cluster. So we could see 6 clusters on our analysis. And we are still working on the data processing to assign the cell types for each clusters. And this is like the data output. So basically, of course, that the software gives you much more than that. So there are a lot of Excel table sheets, but there's also this nice output that gives you like some quality control of your data, and also like a good summarize of what you had. So then you know like the number of cells that you targeted. So for example, I targeted 5,000 cells, and we got like almost 4,000 cells. And this is the number of fragments that we had per cell, and then the fragments that actually overlapped in any targeted region that we expect to be like the region to be accessible, and also like the transposition events, like. And for this, of course, we had some warnings, but what is important to know that you will have this this nice output, and you can look in your data. And again, like this is other part of the output that we have. So we had not only this, but we also have like the nice tCNA graphs showing like your clusters according to the cell characteristics, like to the similarity between them. And here we also have like specific information. So the percentage of your sample that overlap enhancer regions or promoter regions. And this we know that are important data that we can extract from the ATAC-Seq data. So, and then you have like very detailed, and it's just an example because we have much more than that as output results. So we can conclude that it's like an easy machine that we can interrogate relevant biological questions that can advance our knowledge on cell heterogeneity as well as cell states and drug discovery and biomarker screening and discovery. We are seeking to implement the technology in Sao Paulo. So right now, like yesterday, the machine was set up in our lab, and from September to November, we plan to optimize the technology using fresh and cryopreserved tissue. And in December, we are very excited to have our facility opening, and also the technical workshop promotion and training. So we really want to have this running like in a short period of time that a lot of researchers can come and work on this facility. So I would like to thank you for your attention. And here is our contact, and I'll be happy to answer any questions that you might have. Thank you very much.
Hello everyone again. Today we're gonna go over the protocol for the Chromium Controller. This is the first operational machine in Brazil for the Chromium Controller. It just arrived. It arrived yesterday in our lab. We had everything set up, and now we are demonstrating how to use the machine. So we're gonna use a training kit to demonstrate step by step for you. So the first thing that we discussed during our webinar was that there are several important steps for us to get like a good quality sample. And the first step is the sample preparation. So, and the sample preparation will depend on your research question. So here we are going to start like you had already your sample prepared according to the best protocol that you have for your research question, and then we are going to start from that. So the first step is we are going to mix your sample with the master mix, and we are going to vortex and do like several little steps in between, and then we will prepare so master mix with your sample, your oil, and your gel bits, and you're going to put in this gas tank with the chip, and we're going to put to run in the machine. So this is the overview of this training for the Chromium Controller. So this is our holder that we will fit in the machine, and this is the chip. And Lucio, I'm gonna show you, it's very easy. So we're just gonna open this up, and then you see the chip here. Again, as I told you, the chip will depend on the experiment that you're doing. So it will have like a different QR code here, and the machine will automatically recognize this. But first we will insert in our chip in the holder. So like this, it's very simple, you see? And then close it up. There's like this click, and you know that it's well closed. So now we are mixing our master mix with our sample. We have already vortexed this, and now we are centrifuging it. Now we get our sample. So I'm gonna pipette 73 microliters of the master mix and 2 microliters of the sample. Now 2 microliters. Now I'm adding the sample to each of the master mix. And then obviously, if you are like doing different samples, you're gonna change your chip every time. But but here we are just demonstrating, so I'm just gonna go without changing my chip. And now we're closing the lid. We have already mixed our sample with the master mix. So now I'm adding our mixture of sample with the master mix in our chip. And the well one is where we're supposed to pipette our samples. So it's very important that you don't formate bubbles while pipetting. This is a very crucial step for us. So just need to be careful. So you go like at the very bottom of the well, and then you go up like very, very slowly. Then you avoid bubbles.
Right.
I'm adding to well 1, which is the well sample. So now we are vortexing our gel beads for 30 seconds. And then we are removing this from the vortex adapter. Then we are opening this because we have to centrifuge after vortexing. So now we have to centrifuge. So we put this back here, close it up again. Then I am going to pipette, so I'm going to put the gel beads into the wells in the row number 2, which is the gel bead roll. So this is very important that you don't form bubbles while pipetting, because this is a very dense liquid and it's very easy to form bubbles. So it's very, you need to be very careful while pipetting so you don't add bubbles. So then I'm adding 50 microliters of the gel beads in each well. Then you just go very slow. Really slow. So just for us to recapture, first row we added our sample and our master mix, the mixture of our sample and master mix. And then in the row number 2, we just added the gel beads. And in row number 3, we are adding the partitioning oil. I'm adding 45 microliters in each well. So now we have to attach the gasket here. So it's important that the gasket has no dust on it. This is a very important point, because otherwise the dust will go into your sample or your oil or your gel beads, and you don't want to do that. So I'm attaching the gasket. It's always also very easy. So this part is always go like this, and then you just attach like this. And then we can put in the machine. now I am putting the holder in the Chromium controller. So it's very easy. I just touch the screen, and then you see like the menu to open the machine, and then we just attach like this, and then we just press again here, and then it will close. As I told you, the chip has already the QR code on it, so we will automatically recognize which of the protocols that you are running. So then you just have to press play, and then it will take 18 minutes to get ready. And then the gems formation will happen inside this machine. Now it's ending. 2 seconds, 2, 1. The run is completed. So we open. So now we are removing the gasket. And we have to open the holder and place the holder to expose the wells at 45 degrees, like this. So then you might want to check if there's like a difference in the liquids. But now we are fine. And then now I will recover the gems from the row 3, which is the recovery well row. I'm gonna recover 80 microliters and add in a strip to place on ice. You can see that we have an opaque solution, and this is exactly what we expect to have. Now our gems are on ice. You can keep this on ice, but no more than 1 hour. And now I'm adding the recovery agent. So Lucio, I kept, we need to keep on ice for 2 minutes. Right. And then we're supposed to have 2 phases. Let's see what we had. Can you see that? So we have like this 2 phases, like the bottom one, it's pink, and the one on the top, it's like a translucent phase. And then with that, we know that it worked very well. And afterwards, we just collect this phase on the top, and then we go on with the protocol.
Oh, wonderful.
So then you go to the RT-PCR to form your cDNA, or it depends on the protocol that you're doing. If you're just going to do ATAC, you're just amplifying your DNA. So, Lucio, what did you think? It's very easy, right?
So, I mean, the procedure is very easy.
Yeah. The bench work, it's easy.
I think the critical point would be the sample preparation, right?
Yeah.
So,
and I think this is something that we expect to be doing in the next weeks, in the next couple weeks here.
Yes.
We are like trying to optimize our sample prep with different kind of tissue, so we can use the machine with different kinds of projects.
So the idea is this, I mean, the facility now is open and we are waiting for you guys to bring your samples into this launch this technology here in Brazil. So thank you very much, Camila, to teach me. And it was a real pleasure. I mean, you are ready to go. Thank you very much.
Thank you for the opportunity.
It's great. Thank you very much.
Thank you.
So thank you, Camila. Thank you once more. So it was great. Really, I've been learning experimental things like this. For me, is very exciting. I really enjoy. So we have already a question for you, Camila. So there is a question that they ask me, how can you freeze in the MSR your P, B, and C samples? Your MOOPs.
I'm sorry. So first of all, I would like to say that we recorded these videos and this presentation like in the last minute. So I'm very sorry if I made any mistake. So the first thing is that for the question is that depends on where, which step you are asking. So if you are like having your flash blood and then you are freezing in the DMSO and then afterwards you extract the cells and then go with your protocol, then you can do that. But after you extract your cells, then you have to go right away to the single cell separation.
I see. So, so how critical is this? I mean, to froze. I mean, can you use any frozen sample? I mean, there is a special way to froze down the sample. How it works?
Sorry.
For, I mean, to use the samples that have been frozen. So you have you have a specific protocol to froze down the samples, or?
Oh yeah, so it depends on each tissue that you were using. So which tissue that you were using. So if you're using, for example, in our case, we use a postmortem brain samples, and we actually just snap frozen that in -80. So we don't treat. We don't do any treatment before freezing then. I know that they have some suggestions that you can, I saw that you can fix some cells with methanol, but I think it's for cell culture, not for tissue. Again, like it depends. So it really depends on your research questions and the tissue that you're using. Because if you're using like, for example, for us, like we are using postmortem brain samples, we can never have like, at least for the cryopreserved ones, we can never work with the intact cells. We need the nuclei. Yeah, so.
So it's great. Yeah. So you are very lucky, Camila, to have you back, you know, in Brazil. And I mean, and ready to teach us. I mean, so you have already 4 machines in Brazil. You have this one that is already running. We have one machine coming to Unicamp. You have another one in Ribeirão Preto, and you have one that should be starting to put in business, I would say, in 2-3 weeks, from Professor Wilson Araújo in the Garapuava. So I'm very excited. So I heard that you have also a machine running in Mexico and another one running in Argentina. So this is great now. I mean, so you have them. Know that you have already the technology in place. I think the next step, the critical step for us, Camila, it will be that to teach this technology, right? And have very stable protocols. You have to standardize the protocols, right? So how important is to have very good protocols? I mean.
Yeah, so it's like we were discussing this together before. So it's important that it would be nice if we could have like optimization for different types of tissue, like for this initiative for the ATA. Like maybe we could like have in our facility like just already the protocols already established for different types of tissue. So that would be like a nice way to start with. I think we have 2 more questions.
Yes, go ahead. Go ahead. Can you read the question, please?
Yeah. Can you estimate approximately the cost of reagents per sample? So I'm not sure about like the cost for samples, but I had this quote from the company that sells the 10x reagents here in Brazil, and I asked them for a quote for I think 40 samples, and it was $70,000 or something like that. I have to check. So
anyway, I mean, buy reagents in Brazil, and I think it's like this in the whole Latin America. I mean, it's so complicated because you pay so much more to get these kits here. You know, you pay taxes, and the transportation is very expensive because they have to come on ice, so it's a heavy load and so on. So we have another question, Camila, that I like it very much. It's a very good question. I mean, so should everyone buy their own kit or you should share, you know, because I mean, do you agree? I mean, if you can share, you go to reduce the costs, right? So how is possible, Camila? It's possible to buy these kits? For how long can I keep a kit? I mean, how to say, for how long can the kit is ready, is good to use? Can you keep this for weeks, for months? Do you know this?
I'm not sure what I know. So I just found my quote for the ATAC-Seq for 40 samples, and is $24,000 for this. So then you can divide and know like for each case. And for the second question is, it would be nice because each chip comes with like you can run 8 samples per chip. So if you could like organize with other researchers, then you can maybe use the same chip. It depends on how many samples you want to run at once. And also for the sequence, and this is very critical, we were discussing this with Patricia, and if you can add like more like a lot of runs in this, a lot of like different projects in the same run, and it will dramatically reduce the costs for sequencing.
I see. So this machine is in an open platform. I mean, anyone can use. So you're not charging people to use this machine. So University of São Paulo make this possible. So anyone, if you want to use this machine, you just need to organize that. I mean, and you can. So before you need to set up the protocols. But after this, as Camila said, I mean, something like 2 months, right, Camila, to get the protocols stable, begun to work. So. But this machine is not ours. This machine belongs to everybody. So feel free to use if you need. So, but Camila, I want to thank you once more. Okay, thank you very much. I mean, thank you to come back to Brazil and be ready to teach us. I mean, you are very, very happy to have you back. Okay, and you want to keep you here, Camila. Okay.
Okay.
Thank
Thank you again for the opportunity to share with you, and I'll be happy to keep working together and establish this technology for different tissues and for the purpose of the ATA.
Thanks so much, Camila. Thank you very much.